Hello student, welcome again to EPZ Pathshala. I am Dr. Kirti Jain from Delhi Pharmaceutical Sciences and Research University. Today I will talk on the module Theranostic applications, which are the recent trends in biotechnological applications of nanomaterials under the paper Nanobiotechnology. So students, let us see what we are going to learn in this module. In this module, first we will learn about the theranostic and nanotheranostics. Since these are some new applications of the nanomaterials and some new phenomena that we have to learn. So we will learn first what is basically theranostic and what is nanotheranostics. We will discuss why we are developing or why we are discussing or why we are seeing the recent trend or recent advancement in the biotechnological application. So we will discuss the advantages in this module and also the biotechnological applications of these theranostic agents. The theranostic and nanotheranostic. Theranostic includes the two components, that is imaging and the drug delivery. So with the theranostic you can use or you can achieve the co-delivery, and particularly I should say here the simultaneous co-delivery of the diagnostic agents and the therapeutic agents. Nanotheranostic is the application of nanomaterial in these theranostic applications. This could be defined as applications and development of nanocarriers like polymer drug conjugates, lipid drug conjugates, micelles, nanoparticles, dendrimers, carbon nanotubes, quantum dots for controlled co-delivery, including the sustained and targeted delivery of the diagnostic and therapeutic agents. What are the advantages of theranostics? This include the improved diagnostic and therapeutic effects with the fewer side effects, because you are using the some of the targeting device or the some of the homing device that will deliver the diagnostic agent, the therapeutic agent to the diseased sites particularly. So diagnosis and the treatment of disease at early stage could have the maximum chances of a disease being cured or treated. If not possible to cure, you can cure early, you can treat it properly. So to further with this theranostic applications, you can make the treatment of fatal diseases including infectious diseases like AIDS and non-infectious diseases like cancer and cardiovascular disorders less troublesome or the more easy than in comparison with the conventional strategies. To make easy prognosis of disease like cancer, AIDS, diabetes, cardiovascular disease, other disease is also possible with the theranostics. And we can finally, with the advent of these theranostics agents, we can improve the quality of life for the patients. In this slide you can see I have again shown that why this targeted therapy is important, or why we are saying that it could lead to the improved therapeutic index because of this targeted component. So now the next question is that how we can achieve the theranostic applications with the nanomaterial, or how we can achieve the theranostic application. So this we could achieve by using advanced theranostic nanomedicines which have multifunctional nature, like ability to diagnose the disease and deliver the drug or therapy to the diseased cells. As we have learned in the module 15 that quantum dots have this ability. They have intrinsic ability for the imaging or the diagnosis, and they can also be modified for the delivery of the drug or the therapeutic agent to the diseased cells. So for this purpose we need the simultaneous conjugation of the targeting ligand and biomarkers to the nanocarrier, along with the either loading or the conjugation of the diagnostic agents. As you can see in this figure that there are different functionalities has been attached as well as loaded to the nanocarrier. So the red circle is showing the multifunctional nanocarrier, and in this you can see there is a circle that is depicting, blue circle that is depicting the drug molecule or therapeutic agent. So I have seen here three ways in which you can load this drug molecule into the multifunctional nanocarrier. The first is that you can encapsulate it within the carrier As you can see in the center, there are blue circles. You can conjugate it on the surface directly, or you can conjugate it on the surface with the help of some linker. The second thing, you can conjugate the targeting ligand or the biomarker on the surface of the nanocarrier. The red zigzag line is showing the spacer or the hydrophilic linker or the polymer chain used to link the other functionalities to the surface of nanocarrier. And the last is the diagnostic agent. Diagnostic agent either could be loaded into the nanocarrier or could be conjugated at the surface either directly or by the some linking agents. So in this slide, I am giving you the comparison of a drug carrier system for the targeting only and the drug carrier system for the theranostic application. So for the targeting, see, there are only three things are important. There should be one carrier, there is therapeutic agent or a drug within the carrier, and the targeting ligand. There are three components in the drug targeting system. If we are now considering the carrier system for the theranostic applications, so in this we will need the multiple functionalities to be attached to the surface of nanocarrier. So the first requirement for a nanomaterial to be used or a nanocarrier to be used for the theranostic applications, it is necessary that it should have the ability to conjugate or being conjugated with the or being decorated with the multiple functionalities that could be the drug, diagnostic agent and targeting ligand. So although we learned about the liposomes in the previous module, that is module 12 and 13, but here we will take a little introduction of the liposomes, and then we will see how we can use liposomes for the theranostic applications. So basically liposomes are the nano microparticular colloidal carriers or the vesicular system having size in 80 to 300 nanometer or more than that as well. They are spherical vesicles composed of phospholipid bilayers with an aqueous core which form spontaneously. Liposomes has been explored for the various drug delivery applications as we have discussed in previous modules as well. The modified and the multifunctional liposomes. Why we are taking these again? Because these modified and multifunctional liposomes has enabled the development of the theranostic liposomes. So modified and multifunctional liposomes are the two advanced variants for the conventional liposomes. In the modified, you have vesicles like ethosomes and transferosomes. The ethosomes and transferosomes both are composed of lipid structure, but there is a slight difference in their further composition. Ethosomes has ethanol in their structures and transferosomes has surfactants in their structure. So both these ethanol and surfactants gives elasticity and flexibility to ethosomes and transferosomes respectively. The functional or the multifunctional liposomes are important particularly for the theranostic applications because they are derived by surface engineering with the various moieties which facilitates their recognition by the cell surface receptors, which facilitates their use to be used for the drug delivery application and which facilitates their use to be used for the diagnosis and imaging purposes. The multifunctional liposomes are derived by the surface engineering methods with the different moieties as we have discussed earlier, but further these multifunctional liposomes are extensively being explored for targeted delivery using active targeting approaches via receptor mediated endocytosis. The multifunctional liposomes, which enables the application of liposomes into the targeting and the diagnosis applications or in the theranostic application with the simultaneous drug delivery potential and diagnosis potential or the imaging potential. So here you can see this liposomes having the surface that could be conjugated with the multiple functionalities to render the different applications including the theranostic applications. So here again you can see there is comparison of 4 different types of liposomes: conventional, theranostic, pegylated and ligand targeted liposomes. So here we will focus only on the theranostic liposomes shown on the right hand corner. So in the theranostic liposomes you can see it will be a basic liposome composed of bilayer vesicles having phospholipids having polar head groups made up of phosphates and the tail group made up of long carbon chains, okay, having hydrophobic groups. In that the polar head group, inside the polar head groups of the phospholipid, that's shown by the gray circle, are in the inner part, are enclosing the aqueous environment. On the outer side you can see there are the again these polar head groups shown in that gray color, okay, and in between you can see there are the two tails attached to each phospholipid polar head. These are the hydrophobic tails made up of hydrocarbons or the carbon chains, and that's why they are protruding towards each other, and in this way they get away from the aqueous environment and make themselves arranged in the bilayered vesicles. So here you can see that in the theranostic liposomes, the right hand top corner, you can see that targeting ligand can be attached to the surface of the liposomes along with the functionalized imaging agents. So in this you can see the imaging has been functionalized in such a way that it is conjugated to the liposomes, or it is attached or decorated over the surface of the liposomes, as well as it is also carrying some targeting ligand. So we can directly attach a simple imaging agent, or we can also use the targeted imaging agent. Further, we can also use the liposome encapsulating or embedded with the quantum dots for the imaging applications. Now the theranostic liposomes, which is the main part for this presentation particularly. Liposomes have been used in two ways for the theranostic applications. The first way include the entrapment of the nano-sized diagnostic agents like quantum dots, iron oxide nanoparticles and gold nanoparticles and some other imaging agents into the liposomes. Now this entrapment could also be of two types. One, you can encapsulate the diagnostic agent into the core of the liposomes, or you can also embed this diagnostic agents into the lipophilic phospholipid bilayers, as we have seen in the previous image. There have been shown that phospholipid bilayers. Then the second strategy is to conjugate theranostic agents conjugated to liposomes along with the molecular biomarkers for targeting effect. As you can see in the previous image, in that theranostic liposome part, you can see there has been the imaging agents conjugated to the surface of liposome, and there have been one more biomarker or the targeting ligand attached to the liposome surface. In the last point, I have given some examples of liposomes being investigated for the theranostic applications. So liposomes have been conjugated with the targeting ligands like D-alpha tocopherol polyethylene glycol 1000 succinate, TPGS, which is now extensively being explored for the targeting of the particularly anti-cancer drugs, and it has been used for the targeting of different therapeutic moieties like docetaxel, apomorphine, camptothecin, irinotecan, and imaging bodies also like quantum dots, CY5.5 and some other fluorophores also. So along with this TPGS, folic acid has also been used for the targeting of these theranostic liposomes. The next is nanoparticles. Nanoparticles are the colloidal particulate system having size range in the nanometers, ranging from the 0.1 nanometer to 100 nanometer, and more specifically the 10 nanometer to 100 nanometers. There have been different variants of the nanoparticles, that includes the polymeric nanoparticles. Polymeric nanoparticles are made up of polymers that could be of natural or synthetic origins. Then we have solid lipid nanoparticles. Solid lipid nanoparticles includes the lipid, and these lipids are used for the formation of the nanoparticles. In the nanostructured lipid carriers, NLCs, we use solid and liquid lipids or liquid lipids to develop the biphasic nanocarrier system. And the last one includes the conjugation of the drug to the lipid to form the lipid drug conjugates. Nanoparticles are the next candidates which are being explored and investigated for the theranostic applications. The polymeric nanoparticles could be divided into two types, biodegradable and non-biodegradable nanoparticles. Both of these could be coated with the surfactants to reduce the immunological and intermolecular interactions. These are solid lipid nanoparticles, nanostructured lipid carriers, and lipid drug conjugates are the modified versions of these nanoparticles which are based on the lipid matrix. Solid lipid nanoparticles are made up of solid lipids and have been characterized for the properties such as physical stability, controlled release, good tolerability, and protection of loaded drug from the degradation. NLCs and LDCs both have been developed to some of the limitations of conventional solid lipid nanoparticles like drug leakage, large particle size, chances of gelation, and less drug loading. In the NLCs, we use liquid lipid also with the solid lipid to develop that nanoparticulate matrix. So nanoparticles, as you can see in this figure, there have been nanoparticle conjugated with the different functionalities like with the linker, antibody directed. We have discussed about the antibody directed therapy, in which antibody is used to target the delivery system to a particular site in the body. So this could be a targeting agent. We can use targeting ligands like folic acid, mannose, LHRH, and some other peptides for the targeting. Then there is a spacer and fluorescent dye or some other imaging agents like MRI contrast agent or some other imaging like phthalocyanine, etc., that could be used for the targeting with the nanoparticles. And the drug payload either could be conjugated to the surface or could also be loaded to the nanoparticles. Similarly, the imaging agents or the diagnostic agent could also be present in the similar way, either conjugated to the surface of nanoparticles or loaded into the nanoparticles. And in this way, you can use the nanoparticles for the theranostic applications. So now in this slide, we will discuss about the theranostic applications of nanoparticles. Nanoparticles, particularly the polymeric nanoparticles as well as the metal nanoparticles also, have been evolved as promising candidates for the theranostic applications due to their biocompatibility and the less toxicity, because there are many polymers are available which are biodegradable like PLGA, polylactic glycolic acid, PLA, polylactic acid, and caprolactone. There are so many biodegradable polymers are there. So because of that, we can get the biocompatible nanoparticles. The second important property of nanoparticle is that they can protect the encapsulated drug as well as the diagnostic agents from the harsh environment like gastric environment or some acidic environment, or as well as from the some enzymes. And the last most important part is that the polymeric nanoparticles and as well as the other nanoparticles made up of lipids, including solid lipid nanoparticles and nanostructured lipid carriers, have also shown ability for the control and sustained release of the loaded drug and the diagnostic agents. So now biodegradable polymers like PLGA, PLA with TPGS, PLG plus PEG have been explored by the scientists for the theranostic applications. The next advancement in this theranostic application is magnetic nanoparticles. As we have discussed previously also, there is magnetic targeting. So in this, what we do, we apply magnetic field externally to localize the magnetic nanocarrier to a particular site. And for this, we use iron oxide and magnetite or hematite containing the iron oxide. These magnetic nanoparticles has been investigated as theranostic nanomedicine for cancer treatment, either themselves or with other carrier systems like liposomes and dendrimers. Further, in order to improve the stability of these magnetic nanotheranostics, surface coating of surfactant, oleic acid, PEG or dextran has also been investigated to improve their stability as well as to make them long circulatory into the system. The dendrimers are the another molecule that has been used in the delivery or in the theranostic applications. Again, the same thing is there. There are three components, and again there is availability of, as you can see in this figure, there are internal cavities. So these internal cavities and the presence of the surface groups, multiple surface groups and multiple internal cavities. Although in this figure I have shown the third generation dendrimers. If you go beyond third generation, fourth, fifth, sixth generation, you will find there is exponential increase in the number of surface functional groups as well as the internal cavities. So this thing makes dendrimer important in encapsulating as well as presenting themselves as a template for the conjugation of multiple functionalities, and that is the requirement of the theranostic applications. So here again the image, you can see there are multiple functionalities being attached to the surface of dendrimers. Some functionalities are also encapsulated within the cavities present in the surface. So this is the important aspect of dendrimers. Although we have seen this in nanoparticles, carbon nanotubes also, but with the dendrimers, even it is possible you can calculate the number of surface functional groups, and accordingly you can vary different factors into the during the synthesis of dendrimers as well as during their conjugation with the multiple functionalities to develop the dendrimers having different surface functional groups. As we can see in this image, there are three functionalities which are attached to the surface of dendrimers. In this part of the presentation, we will discuss about the theranostic dendrimers, or we can say how dendrimers could be used for the theranostic applications. So the first thing is the presence of sufficient number of branching endpoints, and the cavities along with the spherical shape enable the higher generation dendrimers to deliver simultaneously the therapeutic agents and the imaging agents. Dendrimers have been investigated for simultaneous targeted delivery of therapeutic agents like paclitaxel, doxorubicin with the diagnostic agents such as CY5.5, phthalocyanins, nanocarbon dots, NIR theranostic agents, that is near infrared theranostic agents, etc., using the targeting ligands such as luteinizing hormone, releasing hormone, LHRH peptide, folic acid, somatostatin, and some other targeting ligands. So next one in nanotheranostic application is carbon nanotubes. In carbon nanotubes, again the pristine and functionalized carbon nanotubes belonging to fluorines, and the functionalization is important here. This functionalization is enabling the conjugation of nanotubes with the various functional groups, and that's why they can be used for the theranostic applications. So now these are the different types, and all these types could be evaluated for the drug delivery and targeting as well as for the theranostic applications. The functionalization, I have shown this slide again because this is the main part that is enabling the carbon nanotubes to be able for the theranostic applications. You can see there are different arrows. If we will start with the single wall carbon nanotubes shown on the left hand side of the slide, the functionalization, we have started with the biocompatible polymer, then the attachment of various ligands. So after functionalizing the then carbon nanotubes with the biocompatible polymer, even without functionalizing, if it is a functionalized carbon nanotubes, you can use it simultaneously for the various conjugation that include drug, imaging agent, and the siRNA and some other agents also. In this part of presentation, we will discuss how carbon nanotubes could be used for the theranostic applications. So evolution of various approaches for functionalization of CNTs rendered functionalized carbon nanotubes to be used for theranostic applications. The single wall carbon nanotubes has been explored itself as photoluminescent agents for imaging of tumor as well as NIR absorbers and heaters for photothermal treatment of tumor. We have discussed previously that carbon nanotubes have photoluminescent property. So single wall carbon nanotubes has themselves investigated as imaging agent. Multi-wall carbon nanotubes have also been investigated with different imaging agents for the various theranostic applications. The functionalization of CNTs allows simultaneous conjugation of multiple functional groups to the surface of nanotubes for various theranostic applications. For example, multi-wall carbon nanotubes have been conjugated simultaneously with 4 different functional moieties that including imaging agent, targeting agent, radionuclide, and a drug molecule. In the imaging agents, you can the dye that has been taken was the Alexa flow dye, that is AF488647. Then targeting agent was the folic acid, and technetium 99 isotope was used as a radionucleotide, and the drug molecule that was delivered with this system was methotrexate. This multifunctional multi-wall carbon nanotubes showed controlled delivery of methotrexate mediated by the folic acid receptor, meaning the folic acid receptor or the conjugated folic acid has targeted this multi-wall carbon nanotube system having the targeting agent, imaging agent, radionucleotide, and the drug molecule to the cancer site specifically because of the overexpression of the folate receptors. Further, it also showed optical detectability of the fluorochromes along with the radio traceability of 99 isotope of technetium. Now we will discuss about some of the nanocarriers that has been used for the theranostic applications in the CNS disorder. CNS disorder, that is central nervous system disorder. So scientists have evaluated the different carrier system for the theranostic applications in the disorders related to central nervous system, that includes the brain related disorders. In this you can say the micelles, they have evaluated the fullerenes, they have evaluated the pegylated liposomes, and also the dendrimers and the nanocapsules and nanospheres. The difference between the nanocapsule and nanospheres is that in nanocapsule there is like a capsule type of structure, means there is a reservoir type of system in which the coating encloses the drug or the some other agents within the nanomaterial. Whereas in the nanospheres, the drug is evenly dispersed into the matrix of the polymer or nanometric carrier system. Nanotheranostics, that has been evaluated for this cancer, that could include some of the materials listed here in this table. You can see there are some commercial magnetic nanoparticles, iron oxide, then polymeric nanoparticles, you have liposomes, spions, silicon nanoparticles, dendrimers, then albumin bound paclitaxel, then you have targeting antisense loaded polymers, and dextron containing the iron oxide. All these have sizes in the nanometers, that is varying from the 10 nanometers to 100 or 300 nanometers, and they have been used with the different targeting agents. In this table the scientist has compiled the information on the different receptor targeted carrier system, that includes the HAPG2 for the liver targeting, then we have plasma proteins, then we have selectin, estrogen receptors, folate receptors, and tumor cell bound nucleosomes. So all these has been evaluated for the nanotheranostic application. In the last column you can see biocompatibility and toxicity evaluations. You can see in the first one there is magnetic response was observed and hyperthermia was applied for the imaging and cardiotoxicity. So the problem was here that you need to evaluate this system systematically to minimize the chances of any toxicity. So now in this table again we will discuss about some more theranostic and nanomedicine. The first one is drug polymer conjugates. In this we have HPMA, therapeutic agent copper, and then that is radioisotope of copper, and there is a targeting agent is RGD, that is arginine glycine D-aspartate peptide. So this has been used for the cancer imaging and radio chemotherapy. So three purpose we are solving here. We are imaging the cancer, we are targeting the chemotherapeutic agent, as well as we are also going for radiotherapy. The second is polymeric nanoparticles, that includes the PLA, TPGS nanoparticles. Drug is docetaxel here, and the diagnostic agent is quantum dots, as well as in the third example also with the solid lipid nanoparticles. In this quantum dots has also been used. The second and third, the targeting agent is folic acid and cyclic RGD peptide respectively. Both have been used for the co-delivery of the therapeutic agent or the chemotherapeutic agents and the quantum dots. The fourth example includes the dendrimers. In that, polypropyl imine dendrimers has been used to deliver the therapeutic agent phthalocyanins and with the targeting agent LHRH for the delivery of single theranostic agent. Then there are also examples of liposomes, micelles, gold nanoparticles, carbon nanomaterials that have been used with the different targeting agents like folic acid. There are some peptides and the passive targeting ability for the simultaneous delivery of diagnostic agent and the therapeutic benefit. So students, finally we could summarize that theranostic applications are the recent advancement in the nanobiotechnology that could gives us the opportunity for the simultaneous monitoring of the disease as well as the therapy of a disease, and it is particularly useful in the disease which are considered as fatal and lethal like cancer, AIDS, HIV positive patients, etc. Then successful development of theranostic systems or devices could bring path-breaking advancement in the biotechnological applications of the nanomaterial, because with the advent of such devices, we could treat the different complicated disorders which are currently thought to be untreatable. So, with these applications, we can simultaneously make exact prognosis about any disease while we are also treating it with these theranostic nanomaterials. Thank you.